Okay, welcome to this third video in the key area 1-2. You've already had a look at the fluid mosaic model of the membrane, and then you've looked at the process of diffusion, where substances can get in and out of a cell by passive transport. And in this key area here, we're going to have a look at another type of transport in and out of a cell, and it's called osmosis. Now, osmosis is actually the same, if we just move this over here, as diffusion, except it's involving water. So it's really the diffusion of water in and out of a cell across the membrane. But whenever water is talked about, we always use the term osmosis. So we've got the definition of osmosis down here. It's the special diffusion of water from an area of high water concentration to an area of low water concentration through a selectively permeable membrane and down a concentration gradient. So all the elements of diffusion are still there, that you're moving from an area of high concentration, except this time it's water, to an area of low concentration, again it's water, and its movement through a selectively permeable membrane, and it's also down a concentration gradient. So really what we're just talking about is diffusion of water. Now, when we come to look at diffusion, we do have to think in terms of water concentration. So we need to think of the water concentration in solutions to do that. And if we look at the water concentrations, it's easier to recognise what direction that water is going to move in. So if you've got something which is a high concentrated sugar solution, for example, like the example we've got over here on this side, this one here, you'll see that if there's a lot of sugar in the solution, you have a high sugar concentration, you'll have a lower water concentration. So if we said, say this was a 10% sugar solution, or sometimes they're written as molar solutions, so say we could say this was a 10 molar solution, it's pretty strong, then the more concentrated it is, the lower the water concentration. If on the other hand we had something like a 1% sucrose or sugar solution, or a 1 molar sugar solution, the lower the number, the less sugar there is in it, and therefore the more water that is there. So here we've got a high water concentration and a low sugar concentration, and it's important when we're looking at osmosis to identify between solutions which solution has got the higher water concentration and which solution's got the lower one. So let's go on and look at some examples of diffusion with these wee diagrams I've got here. Here in this situation here, we're imagining that a cell is placed into pure water. So based on what we've been talking about before, looking at water concentrations, that means that out here is the high water concentration, high WC, and inside the cell will be the lower concentration. I'll just write LC. So the surrounding solution here has a higher water concentration. Therefore, the water will move from outside the cell into the cell by the process of osmosis through the selectively permeable membrane down the concentration gradient. If on the other hand we had a solution that was surrounding the cell, and this time it wasn't in pure water, see if I can get, hold on a wee minute here, just 2 seconds. This time it wasn't in pure water, it's in a solution, not that one here, not that one, sorry. It's in a concentrated salt solution. So this time round, outside here, we have got a strong salt solution. So therefore that means that we've got a low water concentration out here There's a lot of salt in the solution, so the water levels are lower, or the water concentration, sorry, is lower. So inside the cell has got a higher concentration. So what happens is that water will move out of the cell into the surrounding solution. If, on the other hand, you've got a situation where you've got equal water concentration inside the cell and outside the cell, so the surrounding solution has got a water concentration equal to in the cell, then you don't get any net gain of loss of water by osmosis. Now, sometimes people sort of think of it that the water isn't moving either one way or the other. You do get movement of water back and forward, but that movement of water will be equal, so you don't get any net gain or any net loss. So if we were thinking of these cells in a situation like this, if we had a cell in an equal water concentration, then the cell wouldn't gain any water, it wouldn't gain, or it wouldn't lose any water, so it wouldn't lose any weight or any mass. If, on the other hand, we were looking at a cell here, and it's in a concentrated salt solution, so it will lose water from the cell, so the cells will lose mass. And in the same way with the cell that's in the pure water solution, it will gain water. And because it gains water, then the cells will gain mass. And that's important sometimes when we come to have a wee look at osmosis questions. So let's have a look at the actual effect of osmosis on plants and on animal cells. And here we've got a plant cell here, and when it's placed in pure water, we've already said that when a cell is placed in pure water, the water will move into the cell. Now, when that happens with a plant cell, the water will move in through past the cell wall is totally permeable. It lets everything in, but past the selectively permeable membrane. And then what happens is that the water moves into the vacuole, and the vacuole starts to swell up, and it pushes against the cell wall of the plant cell. And there's a term that we use for this. The contents like the cytoplasm and everything get pushed out there as well. And the term that we use for this is that the plant cell is described as turgid. Not turbid, that's a totally different word. Described as being turgid. That is, I suppose you could think of it as the cell being quite swollen. The cell doesn't actually burst because the cell wall that is outside of here is very, very strong, and so that prevents the cells from bursting. So that's a plant cell in pure water. If we have a wee look at a plant cell in, say, a strong salt solution or in a strong sugar solution, what will happen is the water will move out of the vacuole, and it causes the vacuole to shrink. And sometimes what happens as well is the actual cytoplasm shrinks away from the cell wall as well. And there's a term that we use for this. The plant cell is either described as being plasmalized, that's this word here, or flaccid. You can use either of these terms. So that's our plant cells in a sugar solution, strong sugar solution, or in pure water. What happens when we put animal cells into water or into a strong salt solution? And this wee diagram here has got it all summarised. When we put something like a red blood cell, for example, into a strong salt solution, then what happens is the cell will lose water by osmosis and the cell starts to shrink. Or there's another word we use for this, and that word is crenate, C-R. Oh, I don't want that to do that. Let me try again. C-R-E-N-A-T-E. E-N-A-T-E, crenate. If you can't remember that word, shrink will do. If the red blood cell is placed in a concentration where it's equal, then again you get no net osmosis and the cell stays the same. Now in our plant cell, when it was placed in pure water, you remember that the cells swelled up and it became swollen and it became turgid, but it didn't burst. Now when you put something like a red blood cell into pure water, there's no cell membrane there, cell wall there, and what happens is as the water moves into the cell again swells up, but this time the cell will burst because there's just the cell membrane there and it's not strong enough to hold the cell together. So the red blood cells burst. And you have to remember the differences between how the animal cell and how the plant cell react in water or in strong salt solutions, and the differences in the names. If this was a plant cell here, the plant cell would be described as flaccid or as plasmolysed. So we're going to have a quick look at some past exam questions that have come up with this sort of thing. And here's one here. It's describing a piece of potato that was cut from a potato tuber and weighed. It was placed in pure water for an hour, then removed, dried and weighed again. And finally it was placed in a concentrated sugar solution for an hour, removed and dried and weighed again. Which line in the table records the results most likely obtained by this treatment? So in this question, to answer it, we have to have a look at which direction the water will be moving in and out of our potato. So we've cut our piece of potato and it's put into pure water first of all. Now if the piece of potato is in pure water, the water will move into the cell and therefore it will gain weight or gain mass because the water moves in. But then it's removed, dried and it's weighed, and then it's placed in a concentrated sugar solution for an hour, and then it's removed, dried and weighed. So this time round it's put into a concentrated sugar solution, so therefore the water will move out of the potato cell into the surrounding sugar solution because it's got the lower water concentration, so the cell will lose weight. So when we come to have a look at the results that are down here, we're going to have to look for results that will match this pattern here. So we've got our first weighing before the potato, before anything's happened, and we've got choices here of 5 grams, 5 grams, or 6 grams, or 5 grams. Now what we need to do is we need to look for a column where the potato on the second weighing has gained weight. So this one here, it's gained weight, and that is in fact the only one where there's been a weight gain, because that's 5, that's lost, and that's lost. So we're saying to ourselves, oh, is it number A? Well, we can double check because we need to look at the next weighing, and in that it should have lost weight. When we look at this result here, it has lost weight. So therefore the answer to this question is A. Now since we're on the topic of exam questions, I'm just going to have a wee talk about the sort of things that can also come up. In that last question we were looking at, I notice I'm sitting here with my head up to see down my glasses, sorry about that. Anyway, you'll not be looking at the video of me. In the last question we were looking at potato cells being put into solutions. In this question here, or this example here, you sometimes get exam questions where they're using the whisking tubing, and you remember we'll have come across that before in our last video, and also hopefully we'll have done some experiments in class with this as well. But in this question that is here, we've got 2 test tubes. In test tube A, we've got inside the whisking tubing bag, we've got a 10% sugar solution. And outside of the whisking tubing bag, we've got a water solution. In test tube B over here though, the 10% sugar solution is in the outside, and the water is in the inside of the viscing tubing bag. So these are model cells, they're called. They're made up to mimic the effect of the cell membrane of the cell, because that's what the viscing tubing does. Now, the bags are washed and dried after they've been filled in order to make sure there's no surrounding solution on either of them and to make sure that it doesn't affect the results. And then they're left, they're weighed, and then they're put into the test tubes. So we've got starting weights for both bags. Then the test tubes are left for 20 minutes, and then the viscing tubing bags are taken out, they're dried, and then they're reweighed. Now they're dried just to make sure that there's not any of the surrounding solution on them that would affect the results. So the question is, which one's going to gain weight and which one's going to lose weight? Well, let's have a look at our wee rules. We have to identify the water concentration, and in this test tube here, test tube 1, the high water concentration is in the outside here of the cell because it's pure water. Therefore, the water is going to move in. So this bag here is going to gain weight, or this model cell. This one here, the 10% sugar solution, is inside here. Oh, sorry, the 10% sugar solution is outside here. The water solution is inside here, so this is the high water solution inside the cell. So therefore, water will move from inside the cell to outside, where there's the higher sugar solution. So therefore, this cell will lose weight. And as I said, you'll sometimes get questions like this where you have to try and work out, you'll have an experiment that's set up and you have to try and work out which of the model cells are going to gain or which ones are going to lose weight. And to do that, you always have to identify where the high water concentration is and where the low water concentration is. One last exam question, an example here. It tells you here that 3 plant cells, P, Q and R, are shown below. Now the first thing to note in this is it's a plant cell, and it's an osmosis question, because it says if all 3 cells were placed in pure water, so we know it's osmosis because it's talking about water, for an hour, what term would be used to describe the resulting appearance of the cells? Now if you think back to the part where we talked about plant cells in water, and if they're placed into pure water, water will move from the pure water, the high water concentration outside, into the cell and then into the vacuole, and the cell will start to swell up, and the term that we use for that is turgid. So make sure in your notes you've got a definition of osmosis, and you've got an understanding of what is meant by high water concentrations and low water concentrations, and that you know the differences between what happens when an animal and a plant cell is placed in a high sugar solution or in a pure water solution. And that's all for osmosis just now.